typically pertains to an embryo whose genome has been edited through in vitro fertilization, mitochondrial replacement therapy, CRISPR, or a variety of other DNA replacement techniques. At this point in time, gene editing is meant to open up a possible avenue for the cure of genetically controlled diseases like cystic fibrosis. The gene or genes that code for the diseases can be edited out of the genome so that the individual will not be affected and that subsequent generations will not be affected by the trait. As the technology associated with gene editing continues to develop, the traits able to be edited will continue to expand. Diseases, physical characteristics, and personality traits could seemingly be edited with the continued progress of gene editing experiments. Moral and ethical issues are arising to try to better understand the technology and how to use it in the future. What is a gene? A gene is essentially the code for what makes you you. So a gene, which doesn't actually look like a gene, comes in a letter format, and these letter formats code for a protein. Ultimately, those proteins that it code for give you specific traits. Of those traits, some of them could be diseases, which is what we are specifically looking at to cure with gene editing therapy. The techniques that's used in gene editing therapy is mitochondrial replacement therapy. So all of your cells, or most of your cells, have mitochondria, which helps produce the energy that your cells need to function. So when you have a cell, you'll have various mitochondria throughout it. And then you'll also have the nucleus in the middle, which contains the DNA that makes you you. So in this DNA, that's basically what gives you the traits of what you look like and whatnot. But the mitochondria also have their own form of DNA. So in diseases that are affected by mitochondria, typically the genetic sequence of the mitochondria is damaged in some way. So with mitochondrial replacement therapy, what they do is they have a host cell that has a nucleus in it, and then normal mitochondria, and then they have a cell that has its own DNA plus the mitochondria, which has damaged DNA. And what they do is they take out the nucleus of this cell that is affected by the disease because of the mitochondria being damaged, and they take out the nucleus of this cell, but then they reinsert the nucleus from this cell into this cell. These cells were from different mothers, so then what you have is this individual grows up to have mitochondria from a different donor, but since the mitochondria are normal functioning, the person will not have the mitochondrial disorder that they would have otherwise had if they would have developed in this cell. So another technique that can be used in gene editing is CRISPR. CRISPR is a little bit different than mitochondrial replacement therapy, which we had also talked about, in that it doesn't only deal with mitochondrial DNA. It can use your normal somatic DNA, which is basically the DNA that gives you most of the traits that you have. So genetic traits are brought about by genes, which we talked about earlier, and genes are ultimately determined by the nucleotides in a sequence. So when you have a disease, there could just be one or two nucleotides that are different, and that causes you to have a trait, which is ultimately a disease. So what CRISPR does is it uses an enzyme, among some other steps. An enzyme's a protein, to snip this genetic sequence, and it snips out those two nucleotides, and then through another process of some other enzymes, gives you a genetic sequence that no longer has the diseased nucleotides. And since you don't have the diseased nucleotides, you do not have a disease in the individual.
This is a Chucky Beat production.
An argument against this is that we would be permanently altering the human genome, and we don't necessarily know how that will affect the future. It kind of could have some unforeseen consequences depending on how that continues to be passed on from generation to generation. Additionally, the methods that are used to edit DNA are not without error. With that in mind, the gene that you intend on editing could not be edited how you had initially intended it to, and that could have some unforeseen consequences that would have to be looked into before deciding to continue on with gene editing. Not all gene editing techniques are heritable to subsequent generations. For example, in mitochondrial replacement therapy, only offspring that are females can pass on the edited genome. This is because mitochondrial DNA is passed on only by a mother. So by that logic, if a male had their mitochondrial DNA altered, they would not pass that on to subsequent generations. So against this you have what's best termed the My Sister's Keeper argument, which is the potential that the technology could be used to have one offspring to save the life of another. And it kind of just brings up the moral question of where do you draw the line of how you use the technology. There are clearly moral and ethical questions that need to be answered in regards to gene editing. However, we believe that at this point the benefits for the therapy outweigh the risks. At this time, we know that the treatments can effectively treat mitochondrial disorders and potentially other disorders. The argument against its use are largely what-if questions. The technology is not currently being used for the purpose of making siblings to be able to donate tissues with matching tissues. Additionally, the technology is not being used to select for traits like personality, height, or intelligence. We can try to imagine the potential impacts of gene editing before they occur, but ultimately, only time will tell how the technology progresses and what will come up in the future. With that in mind, we think that research into gene editing should continue, but as it continues to advance, we think we might need to readdress some of the issues that have been previously brought up.